Hey everyone. In this video, we're going to be learning about stereoisomers and chirality. And the learning objective is by the end of this video, you should be able to identify the chirality centers in a given molecule. So let's talk about what all this means. So in chapter 11, we learned about isomers. Isomers are different compounds that have the same molecular formula. Well, we can actually have subcategories of different types of isomers. And so the first subcategory are structural isomers, and the second category are going to be stereoisomers. Now, we learned about structural isomers previously, and they have the same formula, but they have a different connectivity. The atoms are bonded in a different arrangement. So the example we saw back in chapter 11 here was butane, or you could have methyl propane. And so you can have all 4 carbons in a row, or you could have 3 in a row with a branch. And so these have a different connection pattern. The only thing they have in common is the same molecular formula. Now, stereoisomers have the same formula. They have a similar connectivity, but they differ only in their 3D arrangement. And so this is a lot more subtle to notice. Now, in these two examples, we have carbon in the center here, and he's connected to white, green, red, and yellow. And in the example on the right, he's also connected to white, green, red, and yellow. But the difference is the orientation of those 4 groups around the carbon. So let's talk about chirality centers, also known as stereocenters. A carbon atom that is bonded to 4 different groups is called a chirality center. And so it's important that they are 4 different groups. And when you have 4 different groups, there's 2 possible ways to orient them around that central carbon. You can orient them in a clockwise or a counterclockwise fashion. And so anytime you have 4 groups that are different around a carbon, it's known as a chirality center. And so a molecule that contains a chirality center, or more than one, is considered a chiral molecule. And so the definition of chirality is a molecule that is not superimposable on its mirror image. And so the simplest example for chiral molecules is like your hands, right? They are perfect reflections of each other. But if you try and superimpose them, they don't line up, right? Your thumbs are going to be on the wrong side. Or if I turn my hand around so that my thumbs match up, well, now this is the back of my hand, but this is the front of my hand. So they still don't look exactly the same. So they have all the same parts, but they're facing in opposite directions. And so that same principle applies to molecules. When you have a chiral center, you can have 2 different 3-dimensional orientations, and they are going to be mirror images of each other. And so your textbook goes into agonizing detail about chiral versus achiral, and achiral simply means not chiral. Okay. And so it gives some examples of household items that are chiral versus not chiral. So a pair of gloves are going to be chiral, right? They are mirror images of each other, but they're not exactly the same. 2 baseball bats are not chiral. They're exactly the same. Even if they're mirror images, one is exactly like the other. 2 drinking glasses, not chiral. 2 pairs of shoes, chiral, because they're mirror images, but your left shoe is not exactly the same as your right shoe. They're in opposite directions. And so a new term to describe these mirror images of each other is enantiomers. So stereoisomers that are mirror images are called enantiomers. And so here are 2 enantiomer molecules. And if you try and superimpose them, you see that the green and red spheres don't line up, much like if you try to superimpose your hands, your thumbs are going to be on the wrong sides. So mirror images are known as enantiomers. Now, how can we identify the chirality center in a given molecule? Well, we need to have 4 different groups attached to the same carbon. And so we can consider clusters of atoms as a single group, if you will. So if we're looking at this carbon here, it's bonded to a hydrogen on top, a bromine on the bottom. It's got an ethyl group on the left and a propyl group on the right. And so that is 4 different groups. And so this carbon here in the middle is a chirality center. Now, this can get trickier when you have ring structures. And so if you have a ring structure and you want to know if there's a chirality center, you have to consider the symmetry of the ring. And so let's take a look at this ring here on the left. If I was to draw a dashed line down the middle of it, and we're looking at this carbon here to see if it's chiral, well, it's got a hydrogen, that's 1 group. It's got a bromine, that's 2 groups. And then we can consider each half of the ring as the 3rd and 4th groups. And so we have to ask ourselves, is this half of the ring different than this half of the ring? And if the ring is symmetrical, then these 2 halves are the same. And so it's not chiral. Now, if your ring is asymmetrical, when we draw the dashed line down there, we've got the hydrogen, the bromine, and then this half of the ring is noticeably different than this half of the ring because of that double bond. And so these 2 groups are different, and so this is chiral. So once again, if the ring is symmetrical, it's not chiral. But if the ring is not symmetrical, then it is chiral because this counts as 4 unique groups. Whereas on the left side, these 2 groups are the same. All right, let's do a study check. Identify each molecule as chiral or achiral. So let's look at A here. We've got a chlorine, a methyl, an ethyl, and a hydrogen. Those are 4 different groups. So this one is chiral. All right, let's look at B here. I've got a chlorine, a methyl group, a hydrogen, and a hydrogen. And we'll notice that these 2 groups here are both hydrogens. So this is not 4 different groups. This is only 3 unique groups. So this is not chiral. All right, and C. We've got a hydrogen, a chlorine, a methyl, and a bromine. Those are all different. So this one is chiral. And let's see if we can identify the chirality center in each of these molecules. So this one here on the left, probably going to be the easiest because it's drawn in an expanded form. So we're looking for a carbon that has 4 different groups. And I'm going to suggest that this one here is going to be my chiral center. Because if we look at the 4 different groups, it's got a ethyl group attached, got a chlorine, got a hydrogen, and it's got a methyl group. And so those 4 groups are different, which means that this right here is the chiral center. Okay. Now, when these are drawn in skeleton, it can be less obvious. In skeleton structures, the hydrogens are implied. And so we need to fill in the hydrogen to remind ourselves that there's 4 different groups. So if I'm looking at this carbon right here, we can see that it has 3 bonds already. Carbon always makes 4 bonds, which means that there's 1 hydrogen attached there that's not shown. So if I was to draw that hydrogen in, now we can look at the 4 different groups. And so it's got an OH, a hydrogen, a propyl, and an ethyl. And so those are 4 different groups, which means that that carbon right there is the chiral center. Now, the one on the right. Let's draw in the missing hydrogen. And there's going to be a hydrogen attached right there, which means we've got an H, an OH, and then if we try to draw a line down the middle here, we'll see that this half of the ring is different than that half of the ring. So since that ring is not symmetrical, that means that this carbon right there is in fact a chiral center. So those are the chiral centers in each molecule. 4 different groups attached to the same carbon. All right, so here is Prozac. Prozac has a chirality center. Can you figure out where it is? And so when I gave this question on an exam last semester, a lot of students tried to say that this was the chiral carbon. But if we're looking here, that's a fluorine, a fluorine, and a fluorine. Those are all the same group. And so that can't be the chiral center. So let's draw our attention over here. If I fill in the hydrogen at that position, this carbon right here has several different groups. It's got the H, it's got the benzene ring, it's got all of that right there, and all of that right there. So those are 4 different groups attached to the same carbon, which means that this carbon right here is our chirality center. Now, why do we care about chirality centers? This may seem like an abstract concept, but it's really important in biology. And so biological receptors in your body, like protein receptors, tend to have a very unique 3-dimensional fit. And molecules, particularly pharmaceutical drugs, need to fit that receptor, kind of like a lock and key, right? Every lock has a unique key that fits in it, right? The inside of the lock has a certain shape that fits a certain shaped key. It's the same thing with your body. Your different receptors have a unique shape, and the molecule has to match that. And so let's say this is the receptor in your body, and this is the molecule that you're trying to use. Well, if we have 2 enantiomers here, all the same parts, but connected in opposite ways, like your left hand and your right hand, one version of the molecule will fit the receptor. In this silly cartoon example, we've got a circle, a triangle, and a square. And the circle fits in the circle hole, the triangle fits in the triangular hole, and the square fits in the square hole. But if the orientation of those groups was backwards, it wouldn't fit, because now the square and the circle are on opposite sides. The square won't fit through the circle peg, and the circle won't fit through the square hole. And so this version of the molecule would not fit the receptor. And that's the case we see with pharmaceutical drugs. Oftentimes, only one enantiomer will fit and the other one won't. And so let's take a look at a classic example of thalidomide. So thalidomide, shown here, was a drug marketed in Europe in the late 1950s, early 1960s. And it was an anti-anxiety medication and they prescribed it to pregnant mothers to cure morning sickness and general anxiety associated with their pregnancy, which worked well, except for the fact that only one version of the drug was the anti-nausea version and fit the receptor, but the enantiomer actually caused birth defects. And they didn't realize this until 9 months after they'd prescribed it when the babies started being born. And at first, they were noticing these babies were being born with severe defects, but they didn't know what was causing it. And it took them a couple years to realize that it was the drug that they had been prescribing. And so for a handful of years, they'd been prescribing this drug to thousands of pregnant women, and the results were catastrophic. It led to severe malformations of limb structure. And so like the arms of these kids wouldn't develop fully. And so you can see it was pretty bad. There were over 10,000 children affected by this drug before it was taken off the market. Thankfully, there was a chemist in America who worked for the FDA, and he banned it from being allowed in the US. But in Europe, it was a huge problem in the early 60s. So this is one of the worst examples of a pharmacology disaster in modern medicine. And so this just illustrates the importance of one chirality center versus the opposite chirality center, right? Only one version fits the receptor. The other version could be disastrous. Okay? So chirality is very important. Some other examples of drugs that have chiral centers are Aleve. So here's the structure of Aleve, and you can see that there's a chirality center here. The active version is a nice anti-inflammatory agent that helps when you have sore joints. The opposite is a liver toxin. So when they market Aleve, they have to be careful to separate these two versions and only give you this one. Another example is L-DOPA versus D-DOPA. L-DOPA is used to treat Parkinson's disease. And D-DOPA is not only not active as a drug, but it actually gives you bad side effects. It's not necessarily super toxic, but it can give you nausea and some other bad side effects. So this is another drug where they have to separate the two enantiomers because only one is active and the other has bad side effects. And then a really common one is ibuprofen. So ibuprofen has a chirality center, and only one version is active in the body. Thankfully, the other version has no side effects whatsoever, and so they don't bother separating these when they make it in the lab. And so they actually sell it to you as the 50-50 mixture of both because it's cheaper that way. And so actually a lot of drugs that have a chiral center, one version's active, but in most cases, the other version is just inactive and your body just digests it for food. It's only in certain circumstances that the opposite version is dangerous. But it's important to recognize when you have a chirality center and test each one individually. So that's it for this video, and I will see you in the next one. Thanks for watching.